to talk a little bit more about how some of these microorganisms cause pathogenesis. So, some of these organisms are going to release degradative enzymes, so they're going to degrade. Usually they're going to target things like collagenase, hyaluronidase, things like that. It's going to allow it to get in past your cells and into the body. Some of them again make toxins, so they could be exotoxins, which are secreted by the bacterial cells, or endotoxins, which is part of. What's endotoxin part of? What kind of bacteria? Gram-negative. What's the endotoxin part of the polysaccharide? Good. So remember that. That's the only endotoxin that exists. So we will talk about some of these as we go throughout. Just have a general idea that these are some of the enzymes that we'll talk about as we go throughout the semester. Be somewhat familiar or start to become somewhat familiar with what they do. Most of them are literally named after the function. So if you have a hyaluronidase, it's going to break down hyaluronic acid. Hyaluronic acid you find a lot of it in connective tissue. If you have that, such as a root-based strep, then based on hyaluronidase, it can penetrate that connective tissue and cause infection. So that's an exercise every time. This is all bacteria can make like root-based strep. Collagenase, same thing, breaks down collagen. Can get through collagen penetrated into your tissue. Clostridium perfringens does that. Clostridium perfringens is one of the ones that can cause gangrene. We'll talk about that here in a couple of weeks. Let me come back to the bacterial and the kinase. We'll do the white test in here. That's kind of cool. You can see that in that tube. See what it looks like. Neuroamidase, as you say, degrades salicylic acid to intestinal mucosa. If ever I should go into that, we won't really talk about that as much. We definitely talk about the gut acid in the spring. Mollusins cause hemolysis, lysosomal blood cells. They tend to be core-shaped proteins that coat in the red blood cells. Strep and staph make hemolysis in general. Lactinase and phospholipases, those are going to bring phospholipids to open up cell membrane. Clostridium perfringens does that too. Oftentimes, the bacterium that make phospholipases to actually lyse cell membranes are trying to get nutrients from inside of our host cells. One of the nutrients they're trying to get typically is iron, but they can also use the cytoplasmic contents of our cells to get other kind of nutrients too, carbon sources, etc. A lot of those proteins or that enzymes, as you say, tend to be iron related, so they're not really expressed and find it super low in the environment. And the bacterial cells start making these phospholipases to break those blue cells to get the iron. You don't really need to know much about that at this point. That's a lot of times how it works. Don't worry about the heparinase. That contributes to bronchiolitis. We have not talked about what bronchiolitis even is, so we don't worry about that. But that's something that Bacteroides fragilis does. Bacteroides fragilis you find in your gastrointestinal tract. It usually, I usually don't talk about this in here, but much more what can happen sometimes if you have somebody that has a mechanical perforation of the bowel. So say they got a car wreck or something and they had some severe abdominal damage, and or say they came in and they got stabbed, or they were undergoing surgery and the surgeon accidentally nicked the gastrointestinal tract. Bacteroides fragilis and E. coli will flora in your gastrointestinal tract that can escape into your abdominal cavity. Your abdominal cavity shouldn't have any bacteria there. That is a sterile state. If you take a you take a sample from someone's abdominal fluid, you shouldn't get anything back. There shouldn't be anything there. But if there's some kind of damage to the gastrointestinal tract, those things can get out. Bacteroides fragilis and E. coli, if somebody ever has an infection of their abdominal fluid, you're going to find those together. So like if you see E. coli, I guarantee you that Bacterial fragilis, they're somewhere. You just Same thing if you find bacteroides, there's your whole one hanging out with it somehow. They tend to work very sympathetically with one another to cause some really nasty infections. The other place you can see infections like that is if someone has, say someone comes in and you take some abdominal fluid and you see it's growing Bacterial fragilis and E. coli, but they haven't had surgery. They haven't been stabbed or shot or maybe there hasn't been any kind of trauma, haven't been in a car wreck, but they have this infection in their abdominal fluid. That usually points to they have some kind of gastrointestinal cancer. So it's the cancer causing damage to the gastrointestinal tract. Like 9 times out of 10, that's what that's pointing to. Thrombophlebitis is when you have a clot that becomes infected in a vein. You do not need to know that, but that's what thrombophlebitis is. Clot becomes infected in a vein. IgA protease, we kind of talked about that a little already. This table that I pulled out of the textbook, is, to be honest with you, they call it colonizing factor. Why do you think they call IgA protease colonizing factor? What's that mean? Where do you find IgA protease? Let me back up a little bit. Or where do you find IgA? Sorry.
Secretions.
Yeah, secretions, like respiratory tract secretions, things like that. So, why do you think they refer to IgA protease? So, protease breaks down IgA. Why do you think we refer to that as a colonizing factor? You need it to establish the infection. So if you can, eventually, potentially get more viral, good. So if you have IgA, if you have a bacterium like IgA protease, you say it's a respiratory tract pathogen, we have IgA in all those secretions. If you can't get past the IgA to actually attach to your respiratory tract, you're not going to make an infection. And ultimately, eventually, you're not going to be able to make a biofilm either because you never attached in the first place. So they refer to some of these things as colonizing factors because the bacterial cell needs it to colonize. So it just breaks down the IgA. A lot of these pathogens, especially the examples that are given up there, Neisseria meningitis, Haemophilus influenzae, Streptococcus pneumoniae, we've talked about a little bit already. We'll talk more about here in a few weeks. But, well, I'll ask you that too because we talked about it a little bit. Where do you typically find those kinds of infections? Neisseria meningitis, Haemophilus influenzae, and Streptococcus pneumoniae. Where do you?
Respiratory?
Yeah, so they start in the respiratory tract, right? In order to colonize the respiratory tract, you have to make things like colonizing factors like IgA protease actually attach to the respiratory mucosa, and then where can they spread to once they cause a real nasty respiratory tract infection? Some of the names say that too. So they cause inflammatory response in the respiratory tract, they escape, they make their way to the blood-brain barrier, they cause inflammation there, that inflammation causes a breach of the blood-brain barrier, they end up with cerebral spinal fluid. Cerebral spinal fluid is very nutrient rich. So, and there's not a lot of immune cells there either. So bacteria like that a lot. They'll grow like crazy once they get in there. Okay. We'll talk about all these kinds of things as we go through. But I do want to go back up here. Because we'll talk about coagulates and coagulates here over the next week or so. It's coming up. I don't want to order this to you. Okay. Staph aureus. Let's use that example. What it can do is it can make a coagulase. And you can actually see this if you do something called a coagulase test. It'll cause fibrin to clot. What it does is the Staph aureus makes a coagulase and it essentially walls itself off in fibrin. Why do you think it does that? Why Staph on coat itself with a bunch of your fibrin?
Protect itself from the immune system.
Right, because it's not recognize your immune response is not recognized. So it hides out inside this fibrin plot, but essentially it's made itself out of your fibrin. Once the amount of Staph aureus cells reaches a critical number of cells, which how do you think that's determined? We can talk about this a little bit. What's that called? And the bacterial cells can determine how many bacterial cells are form sensing. So the Staph aureus protease does is it hydrolyzes the fiber. So why do you think it wants to hydrolyze the fiber? So there's bread. Yeah. So sometimes people get confused by things like that. Kind of we walk through like that, it seems like real obvious, right? But sometimes people will be like, well, how Staph aureus is making something that makes fiber, but then it's also making something that breaks the fiber down, because it's not making it at the same time. Because if it was making it at the same time, yes, that would be really stupid. It's just not going to work. So it regulates how it expresses the genes for those different enzymes. We'll talk about Staph aureus starting with all the skin soft tissue infections. We'll start to talk about those various factors in those. IgA protease, if so inclined, this is to know this is where it cuts between the constant region and the variable region. Does it really matter where it cuts? Not really. It's degrading the antibody, so it's not going to reflect on it. But if you're interested, that's where it cuts. Again, you see these IgA proteases made by things like Sericoniae and inhibitors, Haemophilus influenzae, strep pneumo, things that are going to have to colonize a site in the body where you find IgA. That's how these organisms affect those things. One way. We talk like this, these are endotoxins for the day, and exotoxins. Exotoxins are made of gram negatives and gram positives. Gram positives tend to be a bunch of exotoxins, but gram negatives, there's not really a difference there. Again, remember, endotoxins are a component of the bacterial cell. They are little polysaccharides, so they are a part of the bacterial cell itself, as opposed to exotoxins are going to be secreted. So what I mean by exotoxins, things like botulinum toxin, botulism toxin, tetanus toxin, diphtheria toxin, those are toxins that are secreted by those microorganisms to do things to our cells and make them infection. We'll talk about some of those as we go through too. Different things that different toxins can do. Obviously, they can allow the bacterial cells to spread through tissues. We talked about that, ironases, collagenases, things like that. They can damage the membranes. If you have a toxin that is damaging the membrane of pretty much any cell, it's technically referred to as a cytolysis. If that virulence factor is only damaging the cell membranes of red blood cells, it's called hemolysin. Can hemolysins damage more than red blood cells? Yeah, definitely. So there's not really a difference. But technically, hemolysin is a type of cytolysin. You don't really need to get into nuance of that, but that's what that's saying. Stimulate production of excessive amounts of cytokines. So we'll talk about that. We talk about like toxic shock syndrome, toxin 1 and 2. So Staph aureus can make that. You typically see it referred to as TSST1. Why is it called TSST1? Because there's also a TSST2. And ultimately what happens is if you have boy, my artistic skills are great. You have an interaction between a major histocompatibility complex, an MHC1, and a T cell receptor. What this TSST1 does is it locks this interaction on, and it keeps it on. And when it keeps it on, what do you think your immune system does?
Stays activated.
It stays activated, it releases a bunch of cytokines. Ultimately, what you end up with is you might end up with a cytokine storm. So you just end up in your immune response going. Toxic shock syndrome, toxin made by staph can do that. A lot of times, the classic example we see in textbooks of this is you have a woman that leaves in a tampon for too long, and it triggers a Staph aureus infection. I should say that's the best word to use, but you get a Staph aureus infection, and then they get this kind of. That's a classic board question. Almost 100% of the time, if somebody that leaves in that sanitary product too long, it can result in this. People are a lot more astute to that than what they used to be, it seems. But it can cause that. So that's called superengineering, toxic shocks and toxic loss. Why superengine? Because it elicits that immunological response that's out of control. It inhibits protein synthesis, like diphtheria toxins, exotoxin, diphtheria toxin, inhibits protein synthesis in your cells. So it inhibits protein synthesis in your cells. You can't make new cells, you can't do metabolism, you can't do lots of things. So it causes cell death ultimately. Activates secondary message pathways, hormone-like effects, alter cell function. There's a ton of different ones to do this. Now I might put a picture of this. Vibrio cholerae does, I didn't go. We'll come back to this in a little bit. Vibrio cholerae is makes a cholera toxin that acts like this. So what it does is it causes the amount of cyclic AMP inside your cells to go up. And what that does is it makes water go out of your cells. So if you have a Vibrio cholerae infection in the gastrointestinal tract, when water goes out of your cells, where's that water going? Into the lumen in the gastrointestinal tract, which causes the diarrhea. So that's the toxin that causes that. If you have Vibrio cholerae that doesn't make cholera toxin, it's not going to cause cholera, because literally this is the symptom of cholera. That's what causes it. Yeah.
Do the gram-positive in your normal flora release exotoxins like in your body?
Only the ones that they're going to release. Usually the ones that are going to make exotoxins are going to be the pathogenic ones.
Gotcha.
So the ones that are in the normal flora usually don't make any kind of exotoxins that are going to cause any kind of pathogenesis. They can make exotoxins to compete with other bacteria. But that's not really, in the context we're talking about, it's not the same, but they can compete with one another.
Okay.
I'm trying to think, I don't want to give you an absolute on that. If you have an opportunistic infection, some of your normal flora might carry the genes for those exotoxins, but they typically would not be expressing them as normal flora. However, if they get to the region of the body where they're not supposed to be, in an environment that's different, just a different part of the body, that can trigger a response where those genes start being expressed. Okay. I feel like I answered the question a lot there.
No, that makes sense though. Yeah, I'm just wondering how that interaction works.
Yeah, I mean, if you have something that's normal flora that gets deposited in a part of the body where it's not supposed to be, I mean, within the genome, there can be different toxins. If given the right environment, they can start to make. But I mean, for the most part, your normal flora is pretty benign where it's at because it's not causing any kind of damage to you. But if it gets where it's not supposed to be, then that's a different story. What else? That's second message pathway. We'll talk about some of those a little bit. A lot of times, what it does, what these different microorganisms do, not always, but a lot of the time, they tend to change like cyclic AMP. But there are some exceptions to that. Inhibit release of neurotransmitters. We'll talk about that. We'll talk about things like tetanus and botulism toxin. Modify the cytoskeleton of target cells. That one's kind of cool. So Bacterial fragilis, we talked about here a little bit already, is an example of one of the microorganisms that does that. It actually gets inside your cells and starts rearranging your active cytoskeleton. So not good for your cell, but that's an example of one that does this. The neurotransmitters we'll talk about here in a little bit. We'll talk about over the next month or so. Endotoxins, I feel like I can talk about endotoxins every other slide, sorry. Again, lipopolysaccharin, lipase effect, causes septic shock. Generally not released until death of the bacterial cell because of the print of the cell. It is toxic in high doses, so I don't think I explicitly said that to you. Endotoxins are toxic in high doses. In low doses, they're not toxic. So this is why I typically give you an example of there's a common urinary tract infection from urinary catheter. It's proceeded for a long time, like 2 weeks, like urosepsis. If you catch it quick enough, you can treat those people and they don't have the effects of septic shock because there's not as many bacterial cells there to give antibiotics to lice to release as much lipid A. Yeah. What? There's a bunch of different gastrointestinal illnesses that, yeah, you just throw up like crazy. I mean, not even necessarily, there's bacterial examples, but there's viral examples too. Norovirus is a good viral example. Everybody know what, does everyone, we can get off topic, I mean, back to house, whatever. Norovirus, everybody knows what norovirus, what's norovirus? Yeah. You're thinking hantavirus?
Yeah.
Yeah. So norovirus is the one you sometimes see in the news like cruise ships. So there's a type of norovirus called norovirus that's named after Norwalk, Ohio, because that's where they found one of the whatever. Doesn't matter. It makes you puke like crazy while you're simultaneously having diarrhea at the same time. Again, there's a lot of different pathogens that your immunological response to them is to get them out. So you're going to puke things to flush them out. Norovirus, when you have people in a confined space, it actually has to go into somewhat aerosolized. You know, people throw up, they puke so hard that some of the norovirus can aerosolize from the puke particles and travel around. Someone else ingests it, and they get norovirus. You can transfer on fomites and surfaces. If you're on a cruise ship in the middle of the Atlantic Ocean, it spreads easy. So you have like the SS poopscapade, like going through the Atlantic Ocean. So this is why sometimes they like quarantine the ships on and then take them out of mission for like 2 weeks, 3 weeks, 4 weeks to disinfect them because that norovirus is all over the place. It makes you extremely sick if you're exposed to it. Like literally simultaneous diarrhea and vomiting at the same time. If you don't know which apparatus in the bathroom you should go into, it's not so pleasant. I'm trying to remember the case. I'm trying to remember where it was at. It was in Philadelphia. I don't know why. I think it was in Philadelphia. There was an apartment complex, and someone in the building had norovirus, and somehow when they flushed the toilet, it got into the sewage pipes in the building and aerosolized out of other people's toilets and like weird, like some of them are weird. This is like why the whole building come down with norovirus. If you have a confined space, that's one of the ones that'll find its way around. So yeah, there's a bunch of them that make you puke like crazy. Because your body's trying so hard to get it out. So norovirus is one of the ones that you just kind of live with it until it goes away. You just realize that.
Here's how that.
Or was it when they had it? I've had that. It is unpleasant. I've seen people that have that. It is unpleasant. Let's go through the professor on that. Don't eat at the cafeteria at the children's hospital. I used to do some things with Nationwide Children's Hospital up in Ohio, and I was hungry, and I was like, let's go to the cafeteria.
The very last one says the LPS not be converted to toxin. Isn't lipid a part of the polysaccharide?
What'd you say?
The very last point and the last bullet point. Not be converted to toxin.
So, okay, let me get kind of autotoxin reducing hormones effect, blah, that. Yeah, so LPS is heat stable. So what's that mean? It means if you try to autoclave it, and it's still there. Yeah, it doesn't do anything to it. So it's heat stable. It's not strongly immunogenic, and it cannot be converted to a toxin. So what's that mean? It means if you take a toxin and you can use like a chemical, say, to activate it, so it kind of unfolds a little bit, so it doesn't recognize whatever target site it's supposed to recognize in you, it causes toxigenic effects. Or you can heat it, so kind of same thing, it destabilizes, so it unfolds it maybe a little bit, so it doesn't have the same toxigenic effects. You can't do that with LPS. So what's that mean? It means you can't take LPS and inactivate it and use it in like a vaccine, or like I said, antitoxin. It's not, you can't. It just doesn't destabilize. So people have asked that a lot of times too. It's just like, well, if we worry about these kinds of E. coli infections and we worry about it causing septic shock because of the LPS, the lipid A of the LPS, why can't we just make some kind of vaccine against like an activated lipid A? No one's figured out how to do that because there's not really been shown a great way to inactivate it. Obviously, you can't dose somebody with lipid A. That would not go well. So you're kind of stuck. So that's all it's saying. It's just changes in the vaccines, toxoid. A toxoid, you talk about toxoids or specific type of vaccination, where you use an inactivated toxin to trigger an immunologic response. This is what LPS looks like. We've kind of talked about this already. You have this lipid A component, you have this core polysaccharide, and then you have these O-specific polysaccharide side chains. Those side chains are oftentimes used to type gram-negatives. Action of endotoxins, a little bit more into how they actually function. Induce release of endogenous pyrogens. What's a pyrogen? And that's something that causes a fever response. That's what a pyrogen is. Interleukin 1, interleukin 6, TNF, prostaglandins, leukotriene. You don't need to know those. Just know that the endotoxin stimulates your immune system to release things that are going to cause a fever. That's what's important. You don't need to know individual things. And we're starting with you, immunology, we're starting to talk about all those common cytokines and everything. Overproduction of many chemical mediators, in addition to those ones there. This is where it starts to get a little kind of tricky. Increased vascular permeability. So what would you do if somebody came in and they had increased vascular permeability? What are you worried about with that? You're worried about them bleeding out, right? So you would give them something that's going to clot. You go down to the next one, initiates complement and blood coagulation cascades. So it can cause coagulation too. So you can't give them something that makes them clot. So if you're worried about coagulation, you would give them a blood thinner. See where I'm kind of going with how you're kind of stuck here? You don't give them any, you can't, right? There's things you can do to attempt to treat, however, they tend to not be successful because that you kind of have both sides of the spectrum there at the same time, potentially. You're kind of opposed. So macrophage activation products lead to tissue damage, so you have tissue damage from this, and then also you have fever from pyrogens, hypotension potentially because your blood pressure will drop because of the increased vascular permeability, disseminated intravascular coagulation, also referred to simply as DIC, and septic shock. So very, very difficult to figure out how to treat because you have opposite things occurring at the same time. Exotoxins. So that one of the differences between exotoxins and endotoxins, besides endotoxins are part of that renal cell and exotoxins are secreted, is exotoxins are going to be toxic in a tiny amount. Endotoxins you need a buttload of. So you're not going to need a lot of the exotoxin to actually cause the effects. These are secreted a lot of times with gram positives. Again, there's gram negatives that make exotoxins too. There's a ton of examples of that as well. So it's not really a way to differentiate the two. Maybe subclassified as enterotoxins. Enterotoxins you find made in GI pathogens. Neurotoxins you find associated with things that cause like meningitis. And cytotoxins you find associated with those pathogens. Or many pathogens, as you said. A lot of exotoxins might be genetically encoded in the bacterial chromosome. So there's a toxin that's on that circular piece of DNA that has all the sense cell needs to survive. Or it might be on plasmid, which we talked about that a little bit, Gen micro, we talked about that in here too. What's going to be on plasmid, what kind of genes? Yeah, like virulence factors, pili, antibiotic resistance, things that it doesn't necessarily need just to survive on like an LVA plate in here, but the accessory things, yeah, generally. Phage, this is when it gets a little bit more interesting. So there are some pathogens that we'll talk about as we go throughout here that their main virulence factor, sometimes their only virulence factor, is from a bacteriophage being infected with a bacterial virus. If you have a strain of that bacterium that was never infected with that bacteriophage, they don't cause the disease process. So that is called phage conversion. Infection with the phage converted that non-pathogenic microorganism into a pathogenic microorganism. That's what immunology is trying to get at. So if they were those bacterial cells after infection with the bacteriophage or phage converted. There's a few examples of that. It can be modified to produce a toxin as a toxoid as a vaccine. So unlike endotoxins, you can take these exotoxins, generally, you can heat and activate them. You can use chemicals and activate them, and then you can use that as a vaccine. Your body will make antibodies against that toxin. If you're becoming infected with that, or as you said, you're becoming exposed to that bacterium that's making that toxin, you already have antibodies against the toxin, so the toxin will work. There's 5 different mechanisms of action for exotoxins generally. We'll talk about these here individually. Don't memorize these. This is just kind of show you where some of the toxins are. These are some examples of some toxins that alter cellular components. We'll talk about some of these and where you find the gene for the toxin. You see those first 3 are on the bacterial chromosome. Don't worry about alpha-toxin yet. You can find that in the plasmid too. There's a cytotoxin in Pseudomonas aeruginosa. That is a phage toxin. You find that associated with phage. If you come across a Pseudomonas aeruginosa strain that was not affected with that phage, it carries that type 2 cytotoxin, then that Pseudomonas aeruginosa isn't going to make that type 3 cytotoxin. That's all this is trying to show you. Trying to think of some examples that might be more familiar with. Superantigens we talked about, SST1, Staph aureus makes that. That's on the bacterial chromosome, so it's always going to have that. Let's go down to the last one. So strep pyogenes makes erythrogenic toxin A and C. Everybody knows that strep throat, right? Yeah. Strep throat is caused by group A strep. The main member, the only member of group A strep we're going to talk about here is Streptococcus pyogenes. I think we'll talk about this maybe in the first day or two. The strep pyogenes usually gets a strep throat infection. And then a few months later they get strep throat again. And 3 years later they get strep throat again. And every once in a while we'll talk about why does this person keep on getting strep throat? Should they have some kind of immune response to it because they've had it like 3 times now? Like what's going on? There's a bunch of different strains of strep pyogenes. There's at least 23 of them, I think. So the chances of you becoming infected with exactly the same strain all this time is pretty small. But probably you had like strain 1 was picking a number. Strain 1, and then 3 months later you had strain 6, and then 3 years later you had strain 4. It's different flavors, I guess you could say. Sometimes you come across strep pyogenes that's been infected with a bacteriophage that carries these erythrogenic toxins. If you are infected with one of those strains that have been phage converted, I'm trying to think how to ask you this without ever having taught you this. If you have strep throat, what more serious condition can that progress into? I know I'm asking a lot of people. One of the symptoms of scarlet fever is something called strawberry tongue. Literally, it makes your tongue, the patient's tongue, it looks like it has a strawberry in their mouth. They'll also sometimes get a rash that's associated with scarlet fever. That rash and the strawberry tongue, those symptoms are caused by this erythrogenic toxin. So what am I trying to tell you? You can get infected with strep pathogenesis that's never been phase converted with these erythrogenic toxins. Those strains are never, ever going to cause scarlet fever. However, if you were unlucky enough, you get infected with the strep pathogenesis that was phase converted, and you don't seek treatment for the strep throat, that can progress into scarlet fever. That's the only difference between like the strep pathogenesis strains, between the ones that can cause scarlet fever and the ones that can't cause scarlet fever, is literally those two toxins. It's just from being infected with that bacteriophage, because that's where the toxins come from. Does that make sense? We'll talk about scarlet fever here over the next month or so. I was just trying to see if I could, how much I could push you on, but I realize you haven't had that anywhere yet. Yes, so that's an example of phage conversion. We'll talk about that here. Diphtheria toxin is another one. Diphtheria toxin inhibits protein synthesis as made by Corynebacterium diphtheriae. Literally the only thing that makes Corynebacterium diphtheriae cause diphtheria is the diphtheria toxin. That is pretty much the only thing it's got in its arsenal. If you come across a Corynebacterium diphtheriae isolate that was never phage converted to harbor that diphtheria toxin, it doesn't cause diphtheria. It's just a very limited impact. And you see some other examples here too. Shigella dysenteriae causes dysentery. We talked about that in the spring semester. So the Shiga toxin itself is plasmid, encoded. That toxin, no, I'm not that mean this morning. This toxin can bounce around outside of this genus and find its way into toxins like this can find their way into certain kinds of E. coli through a phage converting those E. coli strains to make a Shigella-like dysentery illness. Yeah, we're gonna stop there before I confuse the everything crap out of it. This is something we talked about in infectious diseases. I know a few of you had that in there. There's much different kinds of E. coli. Hemorrhagic or toxigenic. If you're interested in that stuff, we talked about this. All nerve impulse transmission. Tetanus toxins made by Clostridium tetani, botulism toxin by Clostridium botulinum. The botulism toxin is phage converted. So again, that means one of the few things Clostridium botulinum has in its arsenal that actually cause botulism is the botulinum toxin. Come across the Clostridium botulinum that was a phage converted, and it's not going to cause botulism. Pretty much all it's got to offer. Clostridium tetani, plasmid. The difference between these two, tetanus toxin is called tetanus-spasmin. What it does is it causes generalized spastic paralysis. So what that means, it means it goes systemically through your body and it contracts all your muscles. Botulinum toxin is a little different. It causes a descending flaccid paralysis, which means a lot of times you'll get this from canned food, someone ingests the toxin, and the paralysis will move descending. So if you're going from where you consumed it in your mouth, descending down, what do you think the first major muscle that allows you to not die is? You're going through your mouth, down your body. Diaphragm. People stop breathing. The diaphragm can't contract anymore because it's locked, relaxed. You have to watch it, people. So you essentially have to watch it, make sure they're not having difficulty breathing, and if they do have difficulty breathing, you have to make sure that there's in the hospital so you can put them on a ventilator or they will die. You don't see that obviously very often, but when you do see it, especially in the United States, it's usually associated with somebody that canned food on their own at home. And it happens on occasion. I've seen that Again, the intracellular signaling, a lot of these things are gonna target cyclic AMP. There's some examples there, we'll talk about some of those. We've talked about super antigens already. Again, things like toxic shock syndrome, toxin 1, gonna lock that T cell and NHC1 response together, so it just keeps on eliciting this immunogenic response. Results in cytokine storm, ultimately, and it can lead to shock, rash, and autoimmune-like response. Why autoimmune-like response? Because your immune system is like out of control. So it's gonna cause cytokine storm and septic shock. A little schematic that's way better than what I can draw that shows how this happens. It just locks this interaction on, and the cytokines just keep rolling. Okay. So when you talk about some of these toxins individually, so this is how the diphtheria toxin works. A lot of these toxins, you'll see the components of the toxin are labeled as A and B. The way I tell people to remember this is A is the active part of the toxin, and B is the binding part of the toxin. B is what allows the toxin to bind onto whatever target site that the toxin's targeting, and A is what has the response, or causes the response, as you say. So here you see the diphtheria toxin taking in the cell using that B, that binding component, and then that A active component that's going to block protein synthesis. Cells can't make new protein, cells die. Can't divide without making new protein. So that's how that's how Bacterial diphtheria works. This is how tetanus and botulinum toxins work. Clostridium tetani, again, makes the tetanus toxin. Tetanus toxin is sometimes referred to as tetanus spasmin. Blocks the release of inhibitory neurotransmitters, and that results ultimately in muscle contraction. So spastic paralysis, generalized spastic paralysis. Muscles are going to stay locked on and contracted. Clostridium botulinum is going to go in the opposite direction. It's going to be muscle relaxation. So what happens is acetylcholine release is blocked at the motor plate, and then the stimulation is blocked. Again, what's that mean? It means the muscles are relaxed. Unlike tetanus, like I said a little bit ago, tetanus is going to be everywhere. All the muscles are going to be locked on. Botulinum is going to descend, so you can watch it descend down the body. How do you think? How do you think you treat Clostridium botulinum? What would you give somebody? Well, let me back this up. What's happened to effect? If you have somebody that ingests Clostridium botulinum, the botulism toxin, tainted canned food, what's causing this? Is it the Clostridium botulinum, or is it that's actually grown in there, or is it the botulism toxin? So how do you think you treat these people? What do you give them? Give them antitoxin. Give them an antibiotic against the botulism toxin. Would you give them an antibiotic? No, because half the time it's not even there. The only thing left in that canned food is the botulism toxin. And half the time the Clostridium botulinum isn't there, or it's dead. So you would oftentimes give these people, the majority of the time, you give them an antitoxin. Hyperactivation, this is what the cholera toxin looks like. A little bit more complex. You need to know. No. But again, A is going to be the activity, B is going to be for binding against the inside of the cell. It's going to really increase cyclic AMP, and ultimately what that does is a bunch of ions are going to go out, and as the ions go out, the water follows, and that water goes out, it goes into the lumen of your gastrointestinal tract, and causes a ton of very watery diarrhea. Why is it watery and not like diarrhea like you used to see it? Because it's so fast, it just releases all the water from all those cells. There's some strains of cholera, specifically, I can't remember serotype. There was one that was responsible for the cholera epidemic in Haiti. I can't remember which year this was now. But some of those people actually passed away before they even had diarrhea the first time. Like the cholera toxin was so good at being cholera toxin that essentially just emptied all their cell water contents into their gastrointestinal tract. So those are nasty ones for color. What's the O, what I'm referring to?
Plastic?
Yeah, you got it.
I got it. LPS.
The component of the LPS, the oligosaccharide. So that's how you type all of these gram negatives. So if I remember right, I'm digging a little deep here. I think the real nasty color strains are O1 and O139. I'm pretty certain that's right. O1 and O139. Toxic color, I don't remember. I remember one of the ID doctors that I work with on occasion, he was saying it's like within 12 hours, like some of those people didn't have diarrhea. The appearance of the stool, that's something we talk about ID as well. Vibrio cholerae, what it looks like is rice water colored stool. So if you make rice, you look at the water, it's kind of this like milky kind of. It's because like there literally was not enough time to make food. Yeah, I mean, I'm trying to think how else to say it, but essentially, yeah, it's just all of the cells just release all that water and just that's the tube that it ultimately empties out through. So, not good. Let's see. I'm trying to think of what we talked about on here. Exo versus endotoxins. This kind of puts a lot of stuff on the same sort of thing. Exotoxins are polypeptide based, they're protein based. Endotoxins, obviously, are LPS, so they're monosaccharide. Exotoxin, you're going to find a lot of times that for a lot of them, it depends on what the exotoxin is. So the exotoxin can be on plasma, it can be on phage, it can be on bacteriochromosome. One of the main things to remember with the exotoxin is food on phage. It's probably phage converted bacterial strains into having that toxin. Endotoxins is a critical component of the actual structure of the gram-negative cell. It's always going to be on the chromosome. Toxicity, really high for exotoxins, so it takes a little amount to have an effect. Endotoxin doesn't take much at all. The effects you see from different exotoxins are going to vary based on what exotoxin is, which is pretty obvious. But the endotoxins are going to cause the same kind of symptoms because lipid A is lipid A is lipid A. It's not really a difference between. It's going to cause fever shock, cytokine storm, things like that. Depending on how high the dose you've got, ultimately it could lead to death, unfortunately. Antigenicity. Again, you can use exotoxin to make toxoid vaccines. You can't use endotoxin to make vaccines. Endotoxins heat stable. The exotoxins are not heat stable, which means if you heat them to a temperature usually about 60 degrees Celsius, that they're going to unfold. You know, even when you cool them down and they refold, they're not going to refold correctly, so they're not going to work anymore. They don't function as a toxin. So you can use a lot of those in vaccines, the toxic vaccines. Examples of exotoxins we talked about there, and endotoxins, again, there's an example up there, meningococcemia. You don't need to know that, just know gram-negatives happen. We'll talk about that. Infection and that sort of thing. We talked about all this already, so I'm not going to try to touch on this too much. Remember the main difference between antigen variation and phase variation. Phase variation is when something's going to be turned on or off, and antigenic variation is going to be when the relative expression is kind of fluctuating with what's being expressed. Antigenic variation is usually going to be for things like villi, the different Bacterial species, outer membrane proteins. Trypanosoma, we want to talk about it here. That's a parasitological infection. We'll talk about that in spring as well. That depends on what paper you read. Then remember the example of phase variation we gave you. Something on or off. Remember what the example I gave you? It's really one of the best examples we can give you because even with the trypanosoma and brucei, like that's kind of on the fence because I've read papers for that for antigenic variation in phase. What's the example of phase variation I gave you? Yeah.
Where the antibody is made for a specific surface protein and then,
Yeah.
Was it Staphylococcus fiber?
What's his last name?
For phase variation,
For-
somehow
Salmonella. phase variation is going to be on or it's going to be off. So what's that mean? It means if salmonella is making FLIC, it ain't making FLJB. If it's making FLJB, it ain't making FLIC. Most other things that we talk about, differences in like surface antigens to escape it, to get around recognizing antibodies, it's just changing the relative ratio of things. So it might make this protein, I should use protein A, I'm going to say protein 1 on its surface, it starts to decrease the expression of protein 1, so it makes a little bit of it, and then it starts to make more protein 2. And then it starts to decrease the expression of protein 2, and then it'll make protein 3. Never really turns off the expression of one of those things or some of those things. It's always making it, it's just changing the relative ratio of how it's making it. So it makes it really hard for your antibodies and for your immune system to recognize those things. There's not a ton of, that's antigenic variation. There's not a ton of examples of phase variation because there's just not a lot like concrete on or off. Salmonella is like the only one I actually know to actually give you as an example because a lot of the rest of them are going to be antigenic variation is going to change the kind of ratios. It's not going to be a burden like I'm turning this one off, I'm making this one. Those don't do that. Capsular antigens and Enterobacteriaceae. So salmonella is in Enterobacteriaceae, it's a member of that. So that's what that's talking about. That's more phase variation. Antigenic drift or shift is a virology example. I typically don't go into that too much. But some of you have virology right now. Lily, what's the difference between drift and shift? You remember? I put you on the spot. Have you done that yet?
No, we haven't.
You haven't done it. Okay. So, influenza virus. You have seasonal flu. Every year you have to get a new flu vaccine. Every year you get a new flu vaccine because whatever strain is causing flu that year likely is the same strain that caused the year before. The reason for that being is you have point mutations in that flu virus. So it changes from year to year. In the United States, or I should say in the Northern Hemisphere, the strain we choose to vaccinate against is whatever strain was the prominent strain in the Southern Hemisphere. Then, in the Southern Hemisphere, they use whatever strain was prominent in the Northern Hemisphere to pack their input their flu vaccine. Because remember, winter is summer, summer is winter, right? So they can use that to figure that out. Does it work? No, not all the time. Sometimes it works well, sometimes we guess wrong, whatever. But those point mutations that just change the strain a little bit where it wouldn't be recognized with the vaccine is called drift. So that's antigenic shift. You have little changes, there's a little bit of a drift that changes the strain a little bit. Antigenic shift is big changes. That's like a reassortment in the genetic material content of influenza virus. So example of that, you have an avian influenza virus, you have a person influenza virus. You have people that live in close proximity to the chickens that have avian influenza. The people flu and the bird flu end up both in the chicken. If they're both in the chicken, they can swap some genetic material between each other a little bit. And you get this kind of hybrid that has some characteristics of the bird avian influenza and some characteristic of human influenza. If that influenza virus has kind of a little mix between avian and human influenza virus gets into a human being, her immune system doesn't know what to do with that because it doesn't even look like a human influenza virus. So what happens oftentimes is those are the kinds of influenza strains that cause pandemics. If you're looking, ultimately that's called antigenic shift, so that's a big shift. If you're looking at people that are young, people that are middle-aged, and people that are elderly, those pandemic strains, which population do they tend to target? Or maybe sickness is a better word. just gonna overreact like crazy to everything because it can't figure out what it is. It's just like, why is Big Bird doesn't make it sick? Like, he doesn't know what to do with it, so your immune response is gonna be like jacked up, like really going. And then those people get really sick. Similar thing happened with some different isolates of SARS-CoV-2, where you had middle aged people who had the most impact because they had the most robust immune line. So again, remember, this is referring to, my laser works, this is referring to phase variation. So that's how things like salmonella, one of the ways, one of the many ways things like salmonella can get around your immune response, it just changes these flagellum genes. Some bacterial cells can make these needle-like structures. That's from a type 3 secretion system. Bacterial cells make this. If I get my lid to stay on. This part down here, see how it's wider down here? This is embedded in the outside of the bacterial cell. And from this, you have this needle-like structure that extends out right here. This will attach to one of your cells and actually penetrate your cell membrane. So if you have your bacterial cell over here, your cell over here, this bacterial cell can make exotoxins, pass them through this needle, and literally inject them into your cells to kill your cells. You see this a lot in gram negatives. You see this with gram negatives. This is kind of a gram negative thing. So it just literally injects the toxin into your cells. Shigella can do this, salmonella can do this. The structure of these type 3 secretion systems are extremely similar to the base of like a flagellum or a motor. So this is what I'm trying to tell you. Like this structure here is real similar to what's at the bottom of the big long flagellum that allows the flagellum to rotate. And they're really going to know that. I don't know why they still do that. But it can be used to inject toxins. E. coli, Yersinia, Pseudomonas, all of these are gram negatives. This is something we find in gram negatives. You don't even know this. However, there are a bunch of protein secretion systems, not just type 3. There's another one called type 6 that uses a structure somewhat similar to this. However, instead of injecting from a bacterial cell into a host cell, like one of your cells, it can inject from a bacterial cell into another bacterial cell. So they can use type 6 secretion systems for competition to kill other bacteria. Similar kind of concept. The type 3 is used to inject things into our cells. So therefore, something we're going to talk about a ton here. Yeah, I want to talk about a little bit in general micro. Siderophores are released by some bacterial cells to gather iron. Or scavenge for iron, you said. Iron you need for different things for bacterial cell. You need it for enzymes that make peptidoglycan. Peptidoglycan will make up the bacterial cell wall. You can't make more peptidoglycan, you need more bacterial cells. You also see iron associated with components of the electron transport chain. If you don't have iron, you can't do electron transport chain of your bacterial cell, which means you can't make ATP, or at least as much ATP as you should be able to make. So those cells don't do well. So what essentially happens is these bacterial cells can, the bacterial cells will make a siderophore, and they will release a siderophore. The siderophore will grab onto free iron. When the siderophore is bound onto iron, there's receptors on the surface of the bacterial cell that will recognize that iron, that siderophore with iron bound, and bring it back into the cell, create the siderophore, and release the iron inside the bacterial cell. The bacterial cell can use iron for all things, we just iron bound. Some bacterial cells don't have genes to make siderophores. Some bacterial cells make one siderophore. Some bacterial cells make 3 different kinds of siderophores. It just depends on what the microorganism is. Where this becomes a little bit of a pickle for us is your body binds up free iron. So bacterial cells don't have access Some of the things it binds up iron in is obvious things like hemoglobin. Heme is the iron-containing molecule, it's the component of hemoglobin. It's bound up inside your red blood cells. Some bacterial cells, obviously, we talked about this too. They make hemolysis and lyse those red blood cells and have a way to get that heme. Transferrin you find in your bloodstream. Binds up iron too. Lactoferrin you find in secretions, breast milk, things like that. Binds up iron. The pickle becomes that the siderophores sometimes have a higher affinity for the iron than these things. And they can actually rip the iron away from these things to take back to the bacterial cell. Your body, and I talked about this, we talked about immune responses. Your body, when you have a fever response, it triggers your liver to release a compound called hepcidin. Hepcidin essentially goes around and binds up any iron that might be potentially free. There are even some siderophores that have a higher affinity for iron than the hepcidin has for iron. It's like, oh, it's nice, it just rips it away from you. So some of these really find a way to get around this level. Bacterial cells are incredibly lazy, as I told you before. They're not going to make the genes for siderophores, siderophore secretions, siderophore receptors, siderophore uptake. They're not going to make any of that stuff unless there's not a lot of iron there. So essentially that gene expression is repressed until the iron goes away, and then that gene expression for all those things turns on, and starts making siderophore, siderophore receptors, siderophore secretions, siderophore uptake. They're not making that stuff unless they're starting to find iron. You do not need to know all this stuff. Just essentially what I just told you. I guess since I literally just gave you an example of this, I might as well show these since it's here. A lot of times the expression of these genes for siderophore production is regulated by something called FIR. FIR is a repressor. FIR is generally a repressor. It stands for ferric uptake. Ferric uptake regulator. And what happens is when it has iron bound to it, it's not repressing. And when it does not have iron bound to it, when there is not iron, it's going to allow genes to be transcribed. When there's not any iron, it's, oh God. It's going to make the genes when there's not iron. It's not going to make the genes when there is iron. Holy God. I can't find words for it apparently. But that's who that FIR. FIR recognizes what's referred to creatively as a FIR box. It's just the reason it's recognizing to want that transcription. It doesn't need to make iron scavenger molecules. Again, some of these microorganisms have a bunch of different ways to do this. This is how Listeria does this. Do you need to know? No, you don't. The endospores, I know we talked about that in Gen Micro. We do endospore staining. What's the two genera of bacteria that make endospores? So, yeah, Clostridium, good. Those are the only medically relevant ones that make Clostridium, Bacillus, or gram-negatives or gram-positives. Positives, no gram-negatives make endospores. At least not any that we care about. Don't tell anybody that's an environmental micro person that they might be kind of angry with that comment. What happens is these bacterial cells come across an environment that's not hospitable for their continued survival. And then they start to sporulate. Say each bacterial cell will make one individual endospore. Endospore production is a way for these microorganisms to survive a stressful environment where there's not water, there's not a herbivores, or there's radiation or whatever. It's to survive through that. It is not a reproductive process. Sometimes students will think it's just like, oh, this bacillus is going to make these endospores and it's going to allow them all to fly. It's just one vegetative cell makes one endospore, and that one endospore, when it finds an environment that's actually a good condition for it to grow in, will become one single bacterial cell again. It's not a multiplicative process. Spores have something inside them called dibenzolic acid. It's coupled with calcium that you also do not need to elements of the endospore incredibly dry. Why do you think the endospores don't have essentially any water or very low amount of water inside of them?
To let them survive for a long time.
When survive longer, if they're exposed to, those endospores are exposed to some kind of radiation, let's say, that oxygen, or not oxygen, that water can be broken down to oxygen free radicals and destroy the endospore. So there's a amount of water there. You know, water that is not making free radicals from. It has intermembrane, two glycan layers that are incredibly thick, out protein coat. Very, very hard to get rid of these, survive for thousands of years. You can take these endospores, throw them in some 100% alcohol. This is easy to throw, never mind. You can throw it very concentrated alcohol, take them back out, put it on a plate, they're going to germinate, and they're going to make more vegetables. They found them 4,000-year-old salt crystals, they found them in sarcophagi, they found them all over the place. They don't really go away. The example I typically give you is if you go into like a lab where they've done a lot of bacillus work, there's been a lot of endospores that's kind of float around in the air, and then you come into it later on down the road and you do some like research, you open your petri dish plate out on the bench, you're probably going to get some bacillus on that, regardless if you want bacillus on it or not. It's just really hard to get rid of that stuff once it exists. This is why I also give the example for all those anthrax flares went through in the early 2000s. The postal facility where the anthrax endospores went through in the mail, they literally built a chlorine dioxide plant next door to the post office and pumped chlorine gas into the post office to get rid of all the endospores. Like once they're there, you get rid of them by fumigating. That's why a lot of the SO3 labs and stuff have a piping system where you can hook up like a gas tank to fumigate. Most of those methods are not real friendly. Obviously, we don't do well with chlorine either. There are some methods to fumigate now that they've come up with where you can like essentially aerosolize nitric oxide. And the only thing it leaves is water and oxygen left when everything's done. If you have electronic equipment in there, everything's going to be shot. So it's, again, really, really hard to figure out. Please don't memorize this. And this isn't even all, but staff makes a lot of greenhouse gases. This is one of the reasons when we go through all this stuff, especially if you have any pathogen variety or you have any chemical variety, for like staph, you go through all these different systems, and we'll talk about like staph aureus, we talk about skin infections, we talk about staph aureus, we talk about respiratory infections and urinary tract infections. And sometimes somebody will be like, why do we talk about this like everything? That's why. Because it has all these different virulence factors and things that gives them the ability to pretty much cause pathogenesis anywhere that it may be. Somewhat true for strep pathogens as well. The repertoire it has to work with is pretty long. So we talk about strep in a lot of places too. They've just over the years evolved to be pretty good at what to do, unfortunately. You know, do not, please do not try to memorize all these factors. Salmonella virulence factors. Salmonella is another one that can cause some very nasty infections. Doesn't make quite as many virulence factors as like a staph aureus, but it has quite a few things to